Hello students, the total length of DNA in human diploid cell is 2.2 meters and the total dimension of human nucleus is 10 to the power of minus 6 meter, that is 0.00006 meters. But we will get astonished, this such lengthier DNA, that is thousand times more lengthier than the nucleus, which is tightly packed inside the nucleus. How it is possible? This is Dr. Mahesh. Today we are going to discuss about the packaging of DNA helix in both prokaryotes and eukaryotes. Based on the nucleus, we are going to classify the cell into two types, prokaryotic cell and eukaryotic cell. Pro means primitive, karyota means nucleus. We can give an example of prokaryote as a E. coli cells or bacteria. Okay. So in bacteria, we can see only naked DNA which is lacking nucleus or nuclear membrane. But in eukaryotes, EU means true, karyota means nucleus. Say for example, in plants and animals, there is a DNA which is present inside the nucleus. In the protoplasm of bacterial cell like E. coli cells, the DNA is always present in supercoiled state. This supercoiled state of naked DNA is called nucleoid. This nucleoid is abruptly present in the protoplasm of the bacterial cell. DNA always exhibits negative charge due to the presence of phosphate groups. This negatively charged DNA tightly wrapped around the positively charged basic proteins called prolamines. Prolamines are nothing but non-histone proteins which are present in the bacterial cell. But in eukaryotes, the negatively charged DNA wrapped over the positively charged basic proteins called histones and form a complex structure called nucleosome. The fundamental unit of chromatin is nucleosome. There are 5 classes of histone proteins. They are H1, H2A, H2B, H3 and H4. Among 5 classes of histone proteins, 4 histone proteins are present always in pairs and form a histone octamer at the core of the histone. Histones are rich in basic amino acids like lysine and arginine. Based on the isoelectric point of amino acid, histone particles exhibit positive charge which attracts the negatively charged DNA. About 200 base pair of DNA which is wrapped over the core of the nucleosome and form a nucleosome size of approximately 110 into 60 angstrom. One nucleosome is linked with another nucleosome with the help of a piece of DNA called linker DNA. The length of linker DNA varies from species to species and this linker DNA is attached to H1 histone proteins. The nucleosome chain appears like beads on string under the electron microscope. These nucleosome further coils and condense to form solenoid, then solenoid to supersolenoid, supersolenoid to chromatin, then chromatin to chromatin fiber. This is the order of packaging of DNA helix from nucleosome, nucleosome to solenoid, solenoid to supersolenoid, supersolenoid to chromatin, chromatin to chromatin fiber. This chromatin fiber further coils and condenses at metaphase stage of cell cycle to form chromosome. The exact structure of chromosome can be studied under electron microscope only during metaphase stage of the cell cycle. The packaging of DNA helix at higher level also requires a set of additional acidic proteins called non-histone chromosomal proteins. They are also called NHC proteins. There are 3 types of NHC proteins. One is structural NHC proteins. They are also called scaffolding proteins. The second one is functional NHC proteins. These functional NHC proteins can be present in DNA and RNA polymerase enzymes. The third NHC proteins are regulatory NHC proteins. For example, for this NHC or regulatory NHC proteins are HMG. HMG means high mobility proteins. These are the groups of proteins which controls the expression of genes. Some part of chromatin is loosely packed and less stained. This chromatin is called euchromatin. Euchromatin is transcriptionally active, which helps in transcription process. But in some other part of chromatin, they are darkly stained and highly condensed, but they are transcriptionally inactive. They are not involved in protein biosynthesis. They are called heterochromatin. This is about packaging of DNA helix in both prokaryotes and eukaryotes. You need to understand about the difference between packaging of DNA helix in both prokaryotes and eukaryotes. In prokaryotes, histones-like proteins are present called prolamines, but in eukaryotes, histones are present. There are 5 classes of histones. Both prolamines and histones are basic amino acid and they possess positive charge, but DNA is always negative charge, and hence there is an attraction towards the positively charged basic proteins. And the other difference is in prokaryotes the packaging of DNA is very simple, but in eukaryotes it is complex because it needs additional acidic proteins for the packaging of DNA helix at higher levels. Thank you so much. So this is about the packaging of DNA in both prokaryotes and eukaryotes. Kindly subscribe our YouTube channel, that is Tattvamasi Educademy. I will meet on next video. Thanks again.